Hello students, welcome to Don Bosco PU College. We have studied in previous classes a chapter called Body Fluids and Circulation. In this chapter, we studied a first one is called as blood and its components. The blood consists the components like blood plasma, blood cells and blood platelets. Blood platelets. Then in the previous classes also we studied a one more topic is blood group. The four types of blood group A, B, AB, then O. Next one is the a RH grouping or RH factor. There is a two type of antigen present on the surface of the RBCs. Now we are going to study the next continuing topic called as a coagulation of blood or blood clotting mechanism. Blood clotting mechanism. Okay. When come to the a coagulation of blood or blood clotting. Okay. Blood clotting means if any injury occurs, the blood vessels are able to rupture, releases the blood is oozes outside, then the blood is able to contact with air to form a network like structure called clot, then the blood is able to stop. Such process or mechanism is called as blood clotting. The blood clotting prevents the blood loss or bleeding of blood or hemorrhage. Okay, this is a unique character in the mammals we see. What the blood is comes outside or oozes outside by an injury, then the blood is able to clot or coagulates within a few minutes because in this blood clotting process the injured cells releases certain substances. Those substances from the injured cells able to attract the blood platelets and to form a clot where the site of injury is occur. Okay. Let us study how the blood clotting mechanism is occur or taken place. Then here I already told the coagulation of blood or blood clotting within a few minutes. This is because of the injured cells releases certain substances. Which are the substances? The substances are nothing but one is called as a prothrombin, second one called as a thrombokinase, and then calcium ions, finally fibrinogens. Okay, first one called as a nothing but the prothrombin, second one called as a fibrinogen, and next one is called as a nothing but the calcium ions. See, first one nothing but the a prothrombin, second one called as a thrombokinase. Okay, then another called as a fibrinogen. Okay, and then calcium ions. Okay, prothrombin, fibrinogen, thrombokinase, and another called as a calcium ions. Okay, see, these are the four factor very essential for blood clotting process. Then the mechanism of blood clotting process we are going to understand a theory that is called Best and Taylor theory. According to this theory, the process of blood clotting we understand clearly. By this theory, the blood clotting process involves the a certain substances released by injured cells. Those are nothing but a prothrombin and then a thrombokinase, then fibrinogen, then calcium ions. Okay. Then when come to the first one, prothrombin. Prothrombin. So it is an unstable protein. Unstable protein. This protein is formed by the liver. This protein is formed by liver and present in the blood plasma. Okay, what the prothrombin is there? It is a unstable protein or inactive substance present in the plasma and formed by liver. Formed by liver. What the prothrombin is there? The prothrombin is present primarily a thrombin. Then the thrombin is converted into then the prothrombin is converted into thrombin. Okay, once again, prothrombin is an inactive form of protein. It is converted into active form called as a thrombin. Okay, then this prothrombin, it can easily break down into smaller compounds like thrombin. The thrombin is an active protein, active substance in presence of thrombokinase enzyme and calcium ions. The second substance called as a thrombokinase. Thrombokinase. It is a lipoprotein widely distributed in the tissues. It acts as an enzyme or an activator. Thrombokinase, it is a lipoprotein, act as an enzyme or activator. This thrombokinase major function is to convert the inactive form of prothrombin into active form of thrombin. Okay. Then where in the blood plasma. Then third component is called as a fibrinogen. It is a soluble protein found and dissolved in the blood plasma in concentration form. It is formed in the liver and decreases in concentration during the liver diseases. ಲಿವರ್ ಏನೋ ಡಿಸೀಸ್ ಆಯ್ತಪ್ಪ ಅಂದರೆ ಇದರ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಡಿಮೆ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಇಲ್ಲ ನಾರ್ಮಲ್ ಆಗಿತ್ತಪ್ಪ ಅಂದರೆ ಇದು ಕಾನ್ಸಂಟ್ರೇಷನ್ ಜಾಸ್ತಿ ಇರ್ತಕ್ಕಂಥ ನೋಡ್ತಾ ಹೋಗ್ತೀವಿ. ಫೈಬ್ರಿನೋಜನ್. ಓಕೆ, what the fibrinogens are there? The fibrinogens are converted into fibrin. Okay, the fibrinogens are inactive form and the fibrins are active form. Such fibrinogen is converted into fibrin. This conversion process is carried by thrombins. Okay, thrombin, it able to convert the inactive form of fibrinogen into active form of fibrin. Then fourth substances nothing but the calcium ions. It is the calcium ions are an inorganic constituent present in the blood plasma required to promote blood clotting mechanism. In the absence of calcium ions, the blood does not clot. Okay, then does not clot. Then one more component is very important, that is called as a blood platelets. blood platelets in the blood to release the certain factor which activate the mechanism of coagulation. The blood platelet initiate the blood clotting. The blood platelets able to activate the calcium ions to clot the blood. To clot the blood. When come to the exact how the process of blood clotting is taken place, this concept I am going to understand you in 3 steps. First, first, yes, yes. When there is an injury occur, the blood vessels are able to rupture, the blood is oozes outside. Where the injury is occur, the injured cells are able to release the some a injured cells or injured tissue first able to release the thrombokinase enzyme. Before that, when an injury occurs, the blood vessels are able to rupture, the blood oozes out when contact with the air. Then what happens? The injured cells or tissue able to release the one enzyme called as a thrombokinase enzyme. Is the first step. Second, the thrombokinase enzyme act on prothrombin in presence of calcium ions converts into active thrombin. Second step. See, this is the a thrombokinase. Okay, this is the thrombokinase. Thrombokinase enzyme. So this thrombokinase enzyme in presence of calcium ions, thrombokinase enzyme in presence of calcium ions converts the prothrombin into thrombin. Prothrombin into thrombin. When the thrombins are formed, the thrombins are further catalysis and conversion of soluble fibrinogen into insoluble fibrin threads. That is the third step. This is the third step. What happens? First ಏನಾಗುತ್ತೆ, ಥ್ರಾಂಬೋಕೈನೇಸ್ ಏನು ಮಾಡುತ್ತೆ, ಪ್ರೋಥ್ರಾಂಬಿನ್ ಇದ್ದಿದ್ದು ಥ್ರಾಂಬಿನ ಕನ್ವರ್ಟ್ ಮಾಡುತ್ತೆ ಇನ್ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಕ್ಯಾಲ್ಸಿಯಂ ಅಯಾನ್ಸ್ ಅಲ್ಲಿ. ಸೆಕೆಂಡ್ ಸ್ಟೆಪ್ ಏನಾಗುತ್ತೆ, ಫಸ್ಟ್ ಸ್ಟೆಪ್ ಏನಾಗುತ್ತೆ, ಥ್ರಾಂಬೋಕೈನೇಸು ಇಂಜೂರ್ಡ್ ಸೆಲ್ಸ್ ಇಂದ ರಿಲೀಸ್ ಆಗುತ್ತೆ ಬ್ಲಡ್ ಪ್ಲಾಸ್ಮಾದಲ್ಲಿ. ಆ ಥ್ರಾಂಬೋಕೈನೇಸ್ ಎಂಜೈಮ್ ಏನು ಮಾಡುತ್ತೆ, ಪ್ರೋಥ್ರಾಂಬಿನ್ ಇದ್ದಿದ್ದು ಥ್ರಾಂಬಿನ ಕನ್ವರ್ಟ್ ಮಾಡುತ್ತೆ ಇನ್ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಕ್ಯಾಲ್ಸಿಯಂ ಅಯಾನ್. ಸೆಕೆಂಡ್ ಸ್ಟೆಪ್ ಆಯ್ತು. ಥರ್ಡ್ ಸ್ಟೆಪ್ ಏನಪ್ಪ ಅಂದರೆ, ದ ಥ್ರಾಂಬಿನ್ ಈಸ್ ಕನ್ವರ್ಟ್ ದ ಫೈಬ್ರಿನೋಜನ್. ಓಕೆ, fibrinogen into fibrin. This conversion process is carried by thrombin. Okay, thrombin. When the fibrins are formed, the fibrins are insoluble thread-like structure. They are formed as a network-like structure. That structure entangle the blood cells and form the clot. Such process is called as a blood clotting. Apart from these substances like prothrombin, thrombokinase, calcium ions, fibrinogen. So the blood platelets also very important to initiate the a blood clotting process, as well as vitamin K also very important, because vitamin K is required by the liver for the formation of prothrombin. Prothrombin formation ಆಗಲಿಕ್ಕೆ ಏನು ಬೇಕು, ವಿಟಮಿನ್ ಕೆ ಬೇಕು. ವಿಟಮಿನ್ ಕೆ ಏನು ಮಾಡುತ್ತೆ, ಲಿವರ್ ಇಂದ ಪ್ರೋಥ್ರಾಂಬಿನ್ ನ ಫಾರ್ಮೇಷನ್ ಮಾಡುತ್ತೆ. ಆವಾಗ ಪ್ರೋಥ್ರಾಂಬಿನ್ ಥ್ರಾಂಬಿನ್ ಆಗಲಿಕ್ಕೆ ಹೆಲ್ಪ್ಫುಲ್ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ದ ಲ್ಯಾಕ್ ಆಫ್ ವಿಟಮಿನ್ ಕೆ ಆರ್ the presence of liver, so the lack of vitamin K, it affect the delay of blood clotting process. So ಹಾಗಾಗಿ prothrombin, ಥ್ರಾಂಬೋಕೈನಸ್, ಕ್ಯಾಲ್ಸಿಯಂ ಅಯಾನ್ಸ್, ಫೈಬ್ರಿನೋಜನ್ ಜೊತೆಗೆ ಬ್ಲಡ್ ಪ್ಲೇಟ್ಲೆಟ್ಸ್ ಬೇಕಾಗುತ್ತೆ, ವಿಟಮಿನ್ ಕೆ ಸಮೇತನೂ ಬೇಕಾಗುತ್ತೆ. ಓಕೆ, ಅಷ್ಟೇ ಅಲ್ಲ, ಇಷ್ಟು ಅಲ್ಲದೇನಪ್ಪ ಅಂದರೆ ಬ್ಲಡ್ ಕ್ಲಾಟಿಂಗ್ ಫ್ಯಾಕ್ಟರ್ಸ್ ಸಮೇತ ಬೇಕಾಗುತ್ತೆ. ಆ ಫ್ಯಾಕ್ಟರ್ಸ್ ಸಮೇತ ಕೊಡ್ತಕ್ಕಂಥದ್ದು ಇದು ಯಾವುದು ನಥಿಂಗ್ ಬಟ್ ದ ಬ್ಲಡ್ ಪ್ಲೇಟ್ಲೆಟ್ಸ್. ಸೊ ಬ್ಲಡ್ ಕ್ಲಾಟಿಂಗ್ ಫ್ಯಾಕ್ಟರ್ಸ್ ಸಮೇತ ಬೇಕಾಗುತ್ತೆ. ಓಕೆ, ಸೊ ಇನ್ so this is about the blood clotting process. Blood clotting process. Okay, once again I am going to discuss the blood clotting process, the coagulation of blood or blood clotting. This concept is we are going to clearly understand by a theory called Best and Taylor theory. Best and Taylor theory. When an injury occurs in that region, the blood vessels are ruptured, the blood is oozes out. When the blood is contact with air, within a few minutes the blood able to clot. This process is called as a blood clotting process. What the blood able to clot within the minutes, this is because of some injured cells able to release the substances. The substances like one is called as a thrombokinase, another called as a prothrombin, then fibrinogen, calcium ion, platelets, vitamin K and blood clotting factors. Okay. First step, the blood clotting process. We are going to understand first process is nothing but when the blood is oozes out in an injured place, the blood contact with air, then in the injured cells the thrombokinase enzyme is releases and comes into the blood plasma. This is the first step. Second step, in the blood plasma, the thrombokinase enzyme convert the inactive prothrombin into thrombin. Okay. Next, the third step, the thrombin is able to convert the inactive form of a fibrinogen into active form of fibrin. Or soluble fibrinogen into insoluble form of a fibrin. The fibrins are thread-like or network-like. They are entangled the cells, blood cells or cells, and form a clot to stop the blood. Such process is called as a blood clotting process. What a prothrombin is converted into thrombin. This reaction is carried by thrombokinase in presence of calcium ions. Here the blood platelets able to initiate the nothing but a blood clotting process. The vitamin K is very essential in the liver. What happens? The vitamin K able to forms the prothrombin. What happens? There is some extra information you have to know. In normal blood vessels, the clotting does not occur mainly because there is no free thrombokinase available to convert the inactive prothrombin into active thrombin. In case thrombin is formed within the blood vessels, it is neutralized by another substance called as antithrombin. Notably, blood vessels ನಲ್ಲಿ ಏನು ಬ್ಲಡ್ ಕ್ಲಾಟ್ ಆಗೋದಿಲ್ಲ. ಯಾಕಪ್ಪ ಅಂದರೆ ಅಲ್ಲಿ ಫ್ರೀ ಥ್ರಾಂಬೋಕೈನೇಸ್ ಸಿಗೋದಿಲ್ಲ. ಸೊ ಯಾವಾಗ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೋ ಇನ್ಆಕ್ಟಿವ್ ಪ್ರೋಥ್ರಾಂಬಿನ್ ಇದ್ದಿದ್ದು ಆಕ್ಟಿವ್ ಥ್ರಾಂಬಿನ್, ಅವಾಗ ಮಾತ್ರ ಏನಾಗುತ್ತೆ ಬ್ಲಡ್ ಕ್ಲಾಟ್ ಆಗುತ್ತೆ. ಸೊ ಹಾಗಾಗಿ ಫ್ರೀ ಥ್ರಾಂಬೋಕೈನೇಸ್ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಇರೋದಿಲ್ಲ. ಇಂಜೂರ್ಡ್ ಸೆಲ್ಸ್ ಏನು ಮಾಡ್ತವೆ, ಆ ಥ್ರಾಂಬೋಕೈನೇಸ್ ಎಂಜೈಮ್ ರಿಲೀಸ್ ಮಾಡ್ತಕ್ಕಂಥದ್ದು. ಓಕೆನಾ. ಸೊ ಇದನ್ನು ನ್ಯೂಟ್ರಲೈಸ್ ಮಾಡಲಿಕ್ಕೆ ಇರ್ತಕ್ಕಂಥ ಒಂದು ಸಬ್ಸ್ಟೆನ್ಸ್ ಅದಕ್ಕೆ ಆ್ಯಂಟಿ ಥ್ರಾಂಬಿನ್ ಅಂತ ಕರಿತಾ ಹೋಗ್ತೀವಿ. ಒನ್ ಮೋರ್ ಈಸ್ ಹೆಪಾರಿನ್. is another strong anticoagulant but not effective as its quantity is very low in the blood. Low in the blood. ಹೆಪಾರಿನ್ ಅನ್ನೋದು ಇನ್ನೊಂದು ಆ್ಯಂಟಿಕಾಗ್ಯುಲೆಂಟ್ ಸಬ್ಸ್ಟೆನ್ಸು. ಇದು ಅಷ್ಟು ಎಫೆಕ್ಟಿವ್ ಆಗಿ ನಮ್ಮ ಬಾಡಿ ಬ್ಲಡ್ಡಲ್ಲಿ ಅಷ್ಟು ಇರೋದಿಲ್ಲ, ಲೋ ಕ್ವಾಂಟಿಟಿಯಾಗಿ ಇರ್ತಕ್ಕಂಥದ್ದು. ಬಟ್ ಇನ್ ಕಾಂಬಿನೇಷನ್ ವಿತ್ ಆ್ಯಂಟಿಥ್ರಾಂಬಿನ್, ಹೆಪಾರಿನ್ ಇನ್ಕ್ರೀಸಸ್ ದ ಎಫೆಕ್ಟಿವ್ನೆಸ್ ಆಫ್ ಆ್ಯಂಟಿಥ್ರಾಂಬಿನ್. ಓಕೆ, ಸೊ to prevent the blood from clotting, either citric acid or its salts or oxalic acid or its salts is added. These substances precipitate the calcium salts from the plasma. Such blood can be preserved for a long time and used for transfusion. ನೀವು ನೋಡ್ಬೋದು, ಕೇಳ್ಬೋದು, ಬ್ಲಡ್ ಬ್ಯಾಂಕ್ಗಳಲ್ಲಿ ಬ್ಲಡ್ನಲ್ಲಿ ಏನು ಸ್ಟೋರ್ ಮಾಡ್ತಾರೆ, ಪ್ರಿಸರ್ವ್ ಮಾಡ್ತಾರೆ. ಯಾವ ಥರ ಅಂತಂದರೆ ಅಲ್ಲಿ ಪ್ರಿಸರ್ವ್ ಮಾಡಿರ್ತಕ್ಕಂಥ ಬ್ಲಡ್ ಕ್ಲಾಟ್ ಆಗೋದಿಲ್ಲ. ಏನಕ್ಕಪ್ಪ ಅಂದರೆ ಸಿಟ್ರಿಕ್ ಆ್ಯಸಿಡ್ ಅಥವಾ ಸಾಲ್ಟ್ಸ್ ಅಥವಾ ಆಕ್ಸಾಲಿಕ್ ಆ್ಯಸಿಡ್ಸ್ ಇಂಥವು ಏನು ಮಾಡ್ತೀವಿ ಆ ಬ್ಲಡ್ಡಲ್ಲಿ ಆ್ಯಡ್ ಮಾಡಿ ಇಡ್ತೀವಿ. ಆ್ಯಡ್ ಮಾಡಿದಾಗ ಏನಪ್ಪ ಅಂದರೆ ಅಲ್ಲಿ ದಿ ಸಬ್ಸ್ಟೆನ್ಸಸ್ ಪ್ರಿಸಿಪಿಟೇಟ್ ದ ಕ್ಯಾಲ್ಸಿಯಂ ಸಾಲ್ಟ್ಸ್ ಫ್ರಮ್ ದ ಪ್ಲಾಸ್ಮಾದಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಕ್ಯಾಲ್ಸಿಯಂ ಸಾಲ್ಟ್ಸ್ನ ಪ್ರಿಸಿಪಿಟೇಟ್ ಮಾಡ್ತಾ ಹೋಗುತ್ತೆ. ಆವಾಗ ಬ್ಲಡ್ ಕ್ಯಾನ್ ಬಿ ಪ್ರಿಸರ್ವ್ಡ್ ಫಾರ್ ಲಾಂಗ್ ಟೈಮ್ ಅಂತ ನಾವು ನೋಡ್ತಾ ಹೋಗ್ತೀವಿ. ಓಕೆ, ಸೊ ದಿಸ್ ಈಸ್ ಅಬೌಟ್ ದ ಬ್ಲಡ್ ಕ್ಲಾಟಿಂಗ್ ಮೆಕ್ಯಾನಿಸಮ್. ಸೊ ಇಲ್ಲಿವರೆಗೂ ನಾವು ಬ್ಲಡ್ ಬಗ್ಗೆ ನೋಡ್ತಾ ಹೋದ್ವಿ. ಬ್ಲಡ್, ಇಟ್ಸ್ ಕಾಂಪೋನೆಂಟ್ಸ್ ಲೈಕ್ blood plasma, a circulating fluid. Second, blood cells like erythrocytes and leukocytes, nothing but the RBC and WBC. Third one, nothing but the blood platelets. Okay, blood platelets. ಇದು ಆದಮೇಲೆ ನೆಕ್ಸ್ಟ್ ನೋಡಿರ್ತಕ್ಕಂಥದ್ದು ನಾವು ಬ್ಲಡ್ ಕ್ಲಾಟಿಂಗ್, ಬ್ಲಡ್ ಗ್ರೂಪ್ನಲ್ಲಿ RH ಫ್ಯಾಕ್ಟರ್, ದೆನ್ ಬ್ಲಡ್ ಕ್ಲಾಟಿಂಗ್ ಪ್ರೋಸೆಸ್. ಇಲ್ಲಿಗೆ ಈ ಚಾಪ್ಟರ್ಲಿ ಫಸ್ಟ್ ಕಾನ್ಸೆಪ್ಟ್ ಅಂದರೆ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಬಗ್ಗೆ ನಾವು ಕಂಪ್ಲೀಟ್ ಆಗಿ ಸ್ಟಡಿ ಮಾಡ್ತಾ ಹೋದ್ವಿ. ನೆಕ್ಸ್ಟ್ ಇನ್ ದ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸ್ಟಡಿ ದ another colorless blood called as a lymph, and second concept of this chapter called as a blood vessels, and third is called as a heart. ಸೊ ಇದ್ರ ಬಗ್ಗೆ ನಾವು ಏನು ಮಾಡೋಣ, ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ನೋಡ್ತಾ ಹೋಗೋಣ. ಥ್ಯಾಂಕ್ ಯು.